A housewife used an unripe papaya to prepare pickled papaya. She sliced the papaya and immersed the slice in water and then in the 20% sucrose solution. The housewife observed that when the slices of papaya were in the water, they were turgid and become bigger in size. However, when the slices of papaya were in the sucrose solutions, they become flaccid and sunken. Based on the observations of the housewife, planned a laboratory experiment to determine the concentration of the sucrose solution, which is isotonic to the cell of the cell of the papaya. The planning of the experiment must include the following aspects: problem statement, aim of the experiment, hypothesis, variables, list of apparatus and materials, technique used, procedure of experiment, presentation of data, and conclusion. I will go into details of every aspect and teach you the technique in answering it. First aspect is problem statement. So, before writing a problem statement, we should know what is a problem statement. So, please bear in mind, problem statement, it is a question. So, always end it with a question mark. The experiment plan is intended to answer the problem statement. So, please be clear with the aim of the experiment. So before writing a problem statement, we have to be very clear with the aim of the experiment. So what is the aim of the experiment? Look at the words highlighted. Plan a laboratory experiment to determine the concentration of sucrose solution, which is isotonic to the cell of the papaya. So the aim is to determine the concentration of sucrose solutions. So from the aim, we need to form a problem statement, a questions. So the questions will be like this. What is the concentration of the sucrose solution which is isotonic to the cell of the papaya? Next aspect will be the aim of the experiment. To write the aim of the experiment is very easy. We just need to read the requirement of the questions again and then find out what is the requirement of the questions. So in these questions, the requirement is to determine the concentration of the sucrose solution which is isotonic to the cell of the papaya. So that will be the aim of the experiment. Next aspect will be the hypothesis. Before writing a hypothesis, we need to know what is hypothesis. Hypothesis is actually a supposition or proposed explanation made on the basis of limited evidence as a starting point for further investigation. In short, it is a smart guess, a prediction of the result of the experiment. Hypothesis can be wrong because it haven't been tested, so you can write any hypothesis, but you have to write a reasonable one. So, in this case, the hypothesis can be the concentration of sucrose solution which is isotonic to the cell of the papaya is lower than 20%. Or you can be more specific. 5% sucrose solution is isotonic to the cell of the papaya. Both of the hypotheses are acceptable. There are no right or wrong. So you can write either one. Next, we have to identify the variables. But before identifying the variables, we have to know what technique we're going to use. These two are goes hand in hand. So before listing out the variables, we need to figure out what technique we're going to use. Many students have no idea what is technique use and they find it very difficult to write technique use. But actually, it is just a summary of how to conduct the experiment. You just need to summarize the whole experiment using one sentence, but in the sentence, you have to include how you handle the manipulating and responding variable. That's why I say technique use and variables, they go hand in hand. Alright, here's come to the most difficult part. Writing technique use is easy, but deciding what technique I'm going to use is really hard. So, how am I going to conduct the experiment? To decide on the technique used, we have to go back to the aim of the experiment. So, now look at the aim of the experiment. The aim of the experiment is to determine the concentration of the sucrose solution which is isotonic to the cell set of the cell of the papaya. So, look at the highlighted words. This is the manipulated variables. So first step, from the aim of the experiment, you identify the manipulated variables. And how am I going to handle the manipulating variables? I'm going to use different concentrations of sucrose solution. So you need to repeat the experiment with different concentration of sucrose solutions. For example, 5% of sucrose solutions, 10% of sucrose solutions, 15% of sucrose solutions, and 20% of sucrose solutions. Next is to identify the responding variables. So, to identify the responding variables, we need to look at the aim of the experiment again. To determine the concentration of the sucrose solution, which is isotonic to the cell set of the papaya, we need to know what is the concentration of the sucrose solution, which is isotonic to the cell set. But how are we going to know whether the sucrose solution is isotonic to the cell set? We need to look at the conditions of the papaya slide. So the responding variables will be the condition of the papaya slide. And how are we going to handle the responding variables? There is various ways in handling the responding variables. So these are the examples. We can observe the condition of the papaya slide, see whether it is turgid or flaccid, or we can touch and feel the condition of the papaya slide, measure the mass difference before and after the experiment, and measure the length differences before and after the experiment. Alright, we already identified the manipulated variables and responding variables. So the last one will be the constant variables. In this experiment, the constant variables can be the size of the papaya slice or the duration of the papaya slice immersed in the sucrose solutions. Next, you need to list down the apparatus and materials. And it is better to write apparatus and materials as one item, because if you separate them and you place the apparatus and materials in wrong category, that might be marks deduction. So to avoid marks deduction, it's better to write them as one item, and make sure that you always include the main materials and apparatus used to handle the variables. Apparatus and material use depends on how you handle the variables. So in this experiment, our manipulated variables is the concentration of sucrose solution. That's why I need to use different concentration of sucrose solutions. And then the main materials will be the papaya slide, because we're going to study the condition of the papaya slide after immersed in different concentration of sucrose solutions. Next, in handling the responding variable, it depends on how you're going to measure the condition. You can use ruler to measure its length, or you can use electronic balance to measure its mass. So if you use ruler, then no need electronic balance. If you use electronic balance, then no need ruler. It's either one, but you need to include at least one into your materials or apparatus. Next, you have to control your constant variable. Your constant variable can be the duration of the papaya slide immersed in the sucrose solutions. That's why you need a stopwatch. Or maybe your constant variables can be the size of the papaya slide. So that's why you need knife and ruler to make sure that all the papaya slide is in equal size. Lastly, you can also include filter paper or tissue, because they need to use it to dry the papaya slide before measuring its mass. So you can see that there are some materials and apparatus are highlighted that are the main and the main apparatus and materials, so make sure that you include this in your experiment. Next aspect will be the technique used. When identifying the variables, we already know what technique we're going to use. So when writing the technique used, we just need to summarize it into one sentence. So this is the sample answer. Place 5 slices of equal mass papaya slices into different concentration of sucrose solutions and measure the mass differences of the papaya slice after 15 minutes. In this one sentence, I already included the manipulated variables, which is the different concentration of sucrose solutions, the responding variable, which is the mass difference of the papaya slice, and also the constant variables, equal mass of papaya slice, and also the duration of the experiment. So this is how you write technique use. Next, you need to write down the procedure of the experiment. Procedure of the experiment, which means how you're going to conduct the experiment. But when you're writing the procedure, you must include the method in handling the 3 variables and keep all the procedures short and precise. It is preferable you write 5 to 6 steps, not up to 10 to 20 steps. So only include the major procedures in handling the 3 variables. Next is the presentation of data. When you conduct the experiment, you have to record the result. And how you're going to record your result? Normally, you record your result in a table. So presentation of data, which means you tabulate the manipulating variables and responding variables. And you always leave the responding variables, which means the result, blank, because you did not conduct the experiment, so you just leave it blank. So I'm going to show you a sample of the presentation of data. Alright, here is the sample answer for presentation of data. So you have to include the manipulating variables, which is the concentration of sucrose solutions. If you're using mass as the responding variable, you have to include the initial mass and final mass, and don't forget to write the unit. Presentation difference in mass, this is up to you. You can include it or you can exclude it. But the important one is the final mass if you're using mass as the responding variable. If you're using length, then you use length. If you're touching the papaya slice, you can write the texture of the papaya slice, maybe flaccid or maybe turgid. This is up to you. It also depends on how you're handling the variables. There is no right or wrong, and there is no exact way to present your data. The final aspect is to write the conclusion. Please do not leave it blank, even though you do not conduct the experiment, but you can use your knowledge in the biology to write a sensible conclusion. So in this experiment, the conclusion can be 5% of sucrose solution is isotonic to the cell sap of the cell of the papaya. Hypothesis accepted. You can even write 10% or 15% of sucrose solution. There's no right or wrong because you do not conduct the experiment. As long as you write something, marks will be given. So, you already learned the technique in answering paper 3. Your task is to plan a laboratory experiment to determine the concentration of sucrose solution, which is isotonic to the cell sap or the cell of the papaya. And the planning of experiment must include the following aspect, which means all the aspects I discussed just now. You just use the technique you learned just now and write it out. So, please submit your homework in hard copy and also soft copy. Hard copy, you have to do it into your notebook and pass up once the school reopen. And soft copy, please email to the email address as stated here. Thank you and have a nice day.